Assalamualaikum dear students. I am Dr. Anam Pario and today we are going to learn histology of testes and ducts. The lecture was taken by Dr. Suraya and in this video I am going to describe the microscopic features. I am going to show you the images of the microscopic images of the testes and the ducts present in the male genital system. The male reproductive system, this is the introduction. The male reproductive system comprises of four components. It comprises of a pair of primary sex glands which are called as testes. Okay, so there is a pair of primary sex glands which is called as testes and a system of genital ducts. Now there are various ducts which are present inside the testes as well as outside the testes. They are epididymis, vas deferens, as well as the ejaculatory duct. So there is a system of ducts. Then you have the accessory sex gland. Now this is the accessory sex gland. This is prostate, which is performing a function of accessory sex gland, a copulatory organ which is called as penis. So this is the copulatory organ which is called as penis. So male reproductive system consists of four components: a pair of testes, a system of genital ducts, accessory sex gland, and a copulatory organ. Learning objectives. At the end of the lecture, the student should be able to enlist the male reproductive organ. The student should be able to describe the histological anatomy of testes and duct system. The student should be able to differentiate these two structures under the microscope. Bismillahirrahmanirrahim. Okay. The testes perform both exocrine and endocrine function. The exocrine function of the testes is to produce the male gametes which are called as spermatozoa, and the endocrine function of the testes is the production of the male sex hormone which is called as testosterone. When we say that testes is a gland, it is of compound tubular variety. So we say that testes is a compound tubular gland as it is having an exocrine function which is production of the spermatozoa. And it is of cytogenic variety. Now you know that secretions are apocrine, merocrine as well as cytogenic. So whole cell is produced by the testes, hence they are of cytogenic variety. So we said that testes are performing exocrine and endocrine function. The testes are compound tubular glands and their secretions are of cytogenic variety. This is a cut section of the testes and I will describe you all the features present in it. When you take a cut section of the testes, each testes is covered by a thick capsule. Each testes is covered by a thick capsule made up of dense, irregular connective tissue. This thick capsule is called as tunica albuginea. Now we will look into the figure. This is tunica albuginea. The tunica albuginea is actually a capsule of the testes which is made up of dense, irregular connective tissue. Posteriorly, in this region, the tunica albuginea is thickened and it projects into the gland as the mediastinum testis. So we said that posteriorly the tunica albuginea thickens and forms the mediastinum testis. So this is the region which is called as mediastinum testis. From the mediastinum testis, thin and incomplete connective tissue septars radiate to reach the capsule. These septars are called as septular testes. Now we will see this in the figure. These are the septular testes. These are the septars which are dividing the parenchyma of the testes into compartments, or you can say into lobules. These are called as testicular lobules. So the septular testes radiate from the mediastinum testis up to the capsule and divide the interior of the organ into approximately 250 pyramidal intercommunicating compartments which are called as testicular lobules. So this is a pyramidal shaped testicular lobule which is created by the septular testes. These are the septular testes. So what we have learned up till now is that when you take a cut section of the testes, the outermost covering is a layer of dense irregular connective tissue which is called as capsule, also called as tunica albuginea. The tunica albuginea is thickened at the posterior end and it is called as mediastinum testes. From the mediastinum testes, incomplete septars arise and they divide the parenchyma into pyramidal compartments or into pyramidal testicular lobules. About 250 pyramidal testicular lobules are created by the septular testes. Okay, ji. This is the frontal section of the testes, and in this figure you can see that outermost covering is tunica albuginea. Very good. This is tunica albuginea which is forming the capsule of the testes. In the posterior region, the tunica albuginea is thickened and is incorporated into the parenchyma. This is called as mediastinum testes. From the mediastinum testes, septars are arising and they are dividing the parenchyma into lobules. So these are the septars which are dividing the parenchyma into lobules. Okay, this is a microscopic image of the testes. Hematoxylin and eosine staining of the testes, and in it you can clearly see the outermost covering, the capsule of the testes which is called as tunica albuginea. The tunica albuginea in the posterior region is seen to be thickened and it is called as mediastinum testes. Now I'll tell you what is rete testes. Wait. This thickening of the tunica albuginea is called as mediastinum testes. From the mediastinum, you can see the septars are arising. These are the septars. These are the septars, and they are dividing the parenchyma into lobules. These are the lobuli testes. These are the lobules of the testes. चलें आगे चलते हैं. So we said that septars create compartments, create lobules, and these are called as testicular lobules. Now every lobule, when you look each lobule, it consists of a highly intricated coiled tubule which is called as seminiferous tubule. So every compartment, every lobule is filled by a highly coiled tube which is called as seminiferous tubule. It is called as seminiferous tubule. ठीक है जी. So this is testicular lobule, pyramidal shape, having a highly coiled tubule which is called as seminiferous tubule. Just beneath the tunica albuginea, there is a layer of loose connective tissue containing network of blood vessels, and this layer is called as tunica vasculosa. It is called as tunica vasculosa. This is a microscopic image, histological image of the testes. A cut section has been taken, and in it you can see a capsule called as tunica albuginea. Below this, or beneath is a better word, beneath the tunica albuginea, there is a layer of loose connective tissue containing network of blood vessels called as tunica vasculosa. So this is tunica vasculosa. Okay, this is tunica vasculosa. Posteriorly, the tunica albuginea is thickened to form the mediastinum testis, posterior portion of tunica albuginea. From it, the septa are arising and dividing the parenchyma into the testicular lobules. Every lobule, there are almost 250 lobules, and every lobule has a highly coiled seminiferous tubule. About 1 to 4 tubules, about 1 to 4 seminiferous tubules are present inside one lobule. तो कितनी lobules हो गई हमारे पास? 250 lobules. Every lobule has 1 to 4 seminiferous tubules. We move to the next thing. Now, how the seminiferous tubules are formed? It says that each lobule houses about 1 to 4 highly tortuous seminiferous tubules. These are actually tubules. The individual tubules usually commences as free blind ends, but the neighboring tubules may form anastomosing loops. So we said that seminiferous tubules, they are actually highly coiled tubules with blind ends, and they may be anastomosing with each other. At the apex of the lobule, the seminiferous tubules lose their convolutions and become straight. These short straight segments are known as tubuli recti. So see, these are the straight tubules, also called as tubuli recti. The highly coiled seminiferous tubules at the region of apex lose their convolutions and become straight. The straight tubules are called as tubuli recti. The tubuli recti pass into the mediastinum. The tubuli recti pass into the mediastinum testes and join a network of anastomotic channels which is called as RETE testes. So look, the straight tubules, they are opening into an anastomosing channel which is called as RETE testes, and it is present in the mediastinum testes. So RETE testes is a channel of anastomosing tubules. From the RETE testes, 10 to 20 spirally wound efferent ductules arise. From the RETE testes, about 10 to 20 spirally coiled efferent ductules leave the testes and open into the duct of Epididymis. So these are the efferent ductules They are opening into the head of the epididymis. Okay, ji. Before we move forward, we are going to revise the section. So this is a cut section of the testes. The outermost covering is the capsule. The capsule is called as tunica albuginea. So this is the capsule of the testes. The tunica albuginea is thickened in the posterior part to form the mediastinum testes. From the mediastinum testes, septa are arising, dividing the parenchyma into testicular lobules. There are about 250 testicular lobules. Each lobule houses 1 to 4 highly tortuous seminiferous tubules. These are the seminiferous tubules, which are intricately coiled tubules. They start as free blind ends, and at the apex of the lobule they become straight and are called as tubuli recti. The tubuli recti open into an anastomosing channel of tubules which is called as RETE testes. The RETE testes is present in the mediastinum testes. From the RETE testes, 10 to 20 spirally wound efferent ductules leave and they enter into the head of epididymis. The seminiferous tubules are supported by the intertubular connective tissue. So this is the channel of ducts which is present in the testes. This is a microscopic image of the seminiferous tubule. You can see the tunica albuginea, the capsule of the testes, and you can see the parenchyma or the lobules. They are filled with highly coiled tubules which are called as seminiferous ducts. So these are sections of seminiferous ducts. These are all the lobules are filled with seminiferous ducts. This is a seminiferous tubule. This is a microscopic image in which you can see that the seminiferous tubule at the apex, it becomes straight, and the straight tubules are called as tubuli recti. The straight tubules or the tubuli recti open into the RETE testes, and from the RETE testes 10 to 20 efferent ductules emerge which leave the testes and open into the head of epididymis. The seminiferous tubules. Now we have taking a section at the level of seminiferous tubule to visualize the epithelium. So we found that seminiferous tubules are made up of a modified stratified cuboidal epithelium. This is a convoluted seminiferous tubule and it is lined by seminiferous epithelium. The seminiferous epithelium is actually modified stratified cuboidal epithelium. The epithelium is resting on a thin basal lamina and is surrounded by a layer of connective tissue which is called as tunica propria. So there is a thin basal lamina surrounded by the tunica propria. In this figure you can see this is the stratified epithelium which is resting on a basement membrane, and you can see the layer of connective tissue. Now the epithelium consists of two types of cells, the spermatogenic cells and the SERTOLI cells. The epithelium of the seminiferous tubule consists of two types of cells, spermatogenic cells and the SERTOLI cells. This is a magnified image, a microscopic image of the seminiferous tubules. This is the epithelium, modified stratified cuboidal epithelium. and you can see two types of cells, spermatogenic cells and the SERTOLI cells. These are the spermatogenic cells and these are the SERTOLI cells. The spermatogenic cells, they lie between the SERTOLI cells and form a stratified epithelium. The spermatogenic cells are actually those primitive germ cells which undergo sexual maturation. They divide into various stages of differentiation and finally they form the spermatozoa. And this process of division and maturation is called as spermatogenesis. This process of spermatogenesis starts at the onset of sexual maturity. So in the epithelium of an adult, you will find different types of spermatogonias. Type A, dark spermatogonias, type A, pale spermatogonias, and type B, spermatogonias. So we will look into the figure. Near the basement membrane, we can find the first stage, which is spermatogonium. The spermatogonium divide, they replicate to form the primary spermatocytes. The primary spermatocytes undergo meiosis and they form the secondary spermatocytes. The secondary spermatocytes undergo second meiotic division and they produce the spermatids. So in the epithelium you will find various stages of the spermatogenic cells. Type A, dark spermatogonia, they are called as dark because their nucleus is darkly staining. They divide to form type A, pale spermatogonia. The type A cells, the type A pale have a lightly staining nucleus. Type B spermatogonia, they have dark staining nucleus. So on the basis of their nuclei, they are called as dark and pale. This is an image in which you can see the different types of spermatogonia. Type A, dark staining. See, their nuclei are dark staining. Type A, pale staining. Their nuclei are comparatively lighter than the dark staining. This is a series of division, the various stages of the spermatogenic cells. Type A spermatogenic cells, spermatogonias, stem cells, they divide to form type B spermatogonias. The type B spermatogonias undergo mitosis and they form primary spermatocytes. Primary spermatocytes undergo the first meiotic division and they form secondary spermatocytes. The secondary spermatocyte undergoes second meiotic division to form the spermatids. The spermatids undergo a process of spermiogenesis. They lose their cytoplasm and they finally form the spermatozoas. So this is a magnified view of the epithelium, of the seminiferous epithelium, and in it you can see the different stages of the spermatogenic cells. Near the basement membrane you can find the most immature cells, which are called as spermatogonia. See, these are the spermatogonias. They are replicating to form the primary spermatocytes. Primary spermatocytes further divide to form secondary spermatocyte and finally they form the spermatids. This is a very beautiful image of seminiferous epithelium. Hope you are understanding the histology of testes. Again, this is a magnifying view of the seminiferous tubule, in which you can see this is the seminiferous epithelium. In the epithelium, there are two types of cells. We said there are spermatogenic cells and there are SERTOLI cells. The spermatogenic cells are present in different stages of maturation. The immature ones are called as spermatogonias. They divide to form primary spermatocytes. They further replicate meiotically to form secondary spermatocytes, and they finally form the spermatids. The spermatids lose their cytoplasm, modify and transform into the sperms. Okay, what is here? Again an etchy staining of testes. Spermatogonias, primary spermatocytes, secondary spermatocytes, and spermatids. These are the spermatids. And in the epithelium, in the lumen, you will find the spores. Now we are moving to the next type of cells which were present in this epithelium. They are called as Sertoli cells. Sertoli cells are also called as sustentacular cells. These are tall columnar cells which are present between the spermatogenic cells. So see into the figure, these are the Sertoli cells. They are tall columnar cells interposed between the spermatogenic cells. They have a large pale staining nucleus located in their basal portion, and their apical portion is filled with lipid and glycogen granules. This is an image of the Sertoli cells. Now you can see these are the Sertoli cells. They are tall columnar cells having a basally located nuclei. Now the Sertoli cells have occluding junctions. Now the Sertoli cells, they are united with other Sertoli cells by the help of presence of occluding junctions. See, these are the occluding junctions which are present in the Sertoli cells. These occluding junctions create two compartments in the seminiferous epithelium. A basal compartment near the basement membrane and an adluminal compartment. The basal compartment contains the pre-meiotic germ cells and the adluminal compartment contains the meiotic population of the germ cells. Functions of Sertoli cells. The Sertoli cells are providing physical and nutritional support to the developing germ cells. By the help of the presence of occluding junctions, the Sertoli cells are forming the blood-testis barrier, which prevents the immune response against the developing germ cells. The Sertoli cells perform the function of phagocytosis. They are performing the function of secretion of anti-Müllerian hormone as well as secretion of androgen binding protein. This is a picture which is showing you the blood-testis barrier. Now this is a large Sertoli cell. See, look at it here. This is a large columnar Sertoli cell, and you can see this is blood-testis barrier. This is a Sertoli cell nucleus. Okay, so this is a Sertoli cell. Now two Sertoli cells are united with each other by the help of occluding junctions, tight junctions. By the presence of these tight junctions, they have created two compartments. Near the basement membrane, there is a basal compartment. So this is the basal compartment. This is the basal compartment. The basal compartment mainly has immature cells, the spermatogoniums and the primary spermatocytes. And this is adluminal compartment. Which compartment? Adluminal, because it is nearer the lumen. So the adluminal compartment mainly consists of cells which have undergone meiosis, the secondary spermatocytes and the spermatids. Now these cells, these haploid cells, do not reach the blood vessels, and an immune response is prevented by this blood-testis barrier formed by the SERTOLI cells. ओके जी, अब हमने क्या कहा था? हमने कहा था, आई एम रिपीटिंग, when you take a cut section of the testes, you find the outermost layer is capsule. Capsule is sending inside septas, dividing the parenchyma into lobules. Every lobule is filled with seminiferous tubule and is supported by intertubular connective tissue. So this is intertubular connective tissue, which is present in the space between the seminiferous tubule. The connective tissue contains collagen fibers, blood vessels, lymph vessels, and nerves. It has different types of cells, including the fibroblasts, macrophages, mast cells, and the interstitial cells of Leydig. Now, look into the figure. This is a seminiferous tubule, and it has connective tissue, which is called as intertubular connective tissue. This is all the space filled by the connective tissue. The connective tissue has blood vessels, nerves, lymphatics, as well as some important cells which are called as interstitial cells of Leydig. So what are Leydig cells? Over here, present in the connective tissue. The Leydig cells are present close to the blood capillaries. These are actually large ovoid cells with a central dark staining nucleus and eosinophilic cytoplasm. So you can see over here, this is a large ovoid cell having a central dark staining nucleus. Now the Leydig cells appear vacuolated under the light microscope, and they are responsible for secreting the testosterone. Their cytoplasm has lipid material, and this lipid is dissolved out during the routine histological appearance, hence they appear vacuolated. They are responsible for the production of testosterone. This is an image in which you can see the intertubular connective tissue containing the Leydig cells. In this one, you can see this is the epithelium of the seminiferous tubule, the seminiferous epithelium having the spermatogenic cells, and this area is the connective tissue. This is the connective tissue region, intertubular connective tissue, having large cells which are called as Leydig cells. The interstitial cells of Leydig, which are responsible for the production of testosterone, the male sex hormone. हाँ जी, ये हो गई हमारी हिस्टोलॉजी ऑफ टेस्टिस. अब हम मूव करते हैं टू द डक्ट सिस्टम. डक्ट्स आर वेरी सिंपल. There are two types of ducts: intratesticular ducts inside the testes. The first is tubuli recti, which are also called as tubules. Then you have retitestes, and you have efferent ductules. So we have three intratesticular ducts. The excretory genital ducts. There is Epididymis and ductus deferens, also called as vas deferens. We are going to see how their epithelium is formed. What is their appearance under the microscope? The first is the tubuli recti. The tubuli recti is formed by the straightening of the seminiferous tubule. Their initial part is lined by SERTOLI cells and their distal part is lined by simple cuboidal epithelium with microvilli. So what you have to memorize, actually they are the straight ends of the seminiferous tubule and their lining epithelium is simple cuboidal epithelium with microvilli. Then you have retitestes. Retitestes is actually anastomotic network of channels. Again, it is lined by simple cuboidal epithelium with microvilli. So it's quite simple. Straight tubule as well as retitestes. Both are lined by simple cuboidal epithelium. This is a seminiferous tubule, and you remember the epithelium of seminiferous tubule? It was modified, stratified, cuboidal. Modified हम क्यों कह रहे हैं? Because it has two types of cells, spermatogenic cells and SERTOLI cells. Spermatogenic cells are present in the various forms of maturation. The seminiferous tubules become straight and they are called as tubuli recti. The tubuli recti, they are lined by simple cuboidal epithelium with microvilli. So this is the epithelium of tubuli recti, simple cuboidal epithelium with microvilli. The straight tubules or the tubuli recti, they open into the retitestes. This is the retitestes. This is the microscopic image of the retitestes. And you can see that retitestes is lined again by simple cuboidal epithelium. This is simple cuboidal epithelium lining the retitestes. From the retitestes, there about 10 to 20 efferent ductules leave the testes, and these efferent ductules are lined by simple columnar epithelium. Ab epithelium change ho gayi. Now the epithelium is simple columnar. In this, there are two types of cells, some non-ciliated principal cells and there are some tall ciliated cells. The efferent ductules, they are surrounded by a thin layer of circularly arranged smooth muscles. This is a microscopic image of the Ductus efferentis, also called as Vasa efferentia, also called as efferent ductules. कोई भी नाम ले सकते हैं. Ductus efferentis कह सकते हैं, Vasa efferentia कह सकते हैं, efferent ductules कह सकते हैं. They are same. It is lined by, this is the epithelium, it is lined by simple columnar cells. Some are ciliated and some are non-ciliated. The efferent ductules open into the head of epididymis. Now epididymis is actually a comma-shaped body, and the epididymis is a comma-shaped body that overlies the posterior superior surface of the testes. It is present on the posterior superior surface of the testes. It has three parts: a head, a body, and a tail. This is head, this is body of the epididymis, and this is tail of the epididymis. The epididymis consists almost entirely of a single complexly convoluted duct which is called as ductus epididymis. It is made up of a single highly convoluted duct called as ductus epididymis. the duct is actually so much coiled. It is so long. It is about 6 to 7 meters long, but it is coiled. Okay, ji. It is a long coiled duct. When you take the cut section of the ductus epididymis, you find that it is lined by pseudo-stratified columnar epithelium. Epithelium भाई फिर change हो गया. In the end, I'll revise all these epithelium. So it is lined by pseudo-stratified columnar epithelium. Look into the figure. This is the pseudo-stratified epithelium of the epididymis. It consists of two types of cells, columnar cells, which are called as columnar principal cells having stereocilia, and it has some small basal cells. These are the stereocilia which can be seen on the apical region of the columnar cells. And in the lumen of the epididymis, you can see the spermatozoas. The lumen of the epididymis contains the spermatozoa. The epididymis is lined by a layer of connective tissue having smooth muscles. So there is a layer of smooth muscles which is present outside the epithelium. The last duct is called as the ductus or the vas deferens. It is a long straight tube having three coats. It has a mucosa, muscularis and adventitia. Now the mucosa is thrown into folds, hence the lumen appears star-shaped. This is the star-shaped lumen of the vas deferens or the ductus deferens because of the folding of the mucosa. The epithelium is pseudo-stratified columnar epithelium. The muscularis is very thick. There is a thick layer of smooth muscles in the ductus deferens, and the smooth muscle is arranged into three layers: inner and outer longitudinal and a middle circular layer. Inner and outer longitudinal, and there is a middle circular layer of smooth muscles. Outside the muscularis, there is a layer of adventitia which consists of connective tissue having blood vessels. Summary. Bismillahirrahmanirrahim. So when you take the cut section of the testes, the outermost covering is formed by a dense, irregular connective tissue which is called as capsule. The capsule is called as tunica albuginea. This is tunica albuginea of the testes, the capsule of the testes. The capsule is thickened posteriorly to form the mediastinum testes. From the mediastinum testes, septas arise, which are called as septular testes, which divide the parenchyma into lobules. These are the lobules, the pyramidal shaped lobules. There are about 250 lobules. Every lobule consists of 1 to 4 highly convoluted seminiferous tubules. When you take the cut section of the seminiferous tubules, their epithelium is called as seminiferous epithelium, and it consists of two types of cells, spermatogenic cells and the SERTOLI cells. The seminiferous tubules are supported by the intertubular connective tissue, and the connective tissue consists of interstitial cells of Leydig, which are responsible for the production of testosterone. Now, the seminiferous tubule, at the apex of the lobule, These lose their convolutions and they become straight tubules, which are called as retitestes, straight regions of the seminiferous tubule. The straight tubules, they open into the retitestes. From the retitestes, efferent ductules emerge and the efferent ductules open into the head of epididymis. The epididymis has a head, a body, and a tail. This is the tail of the epididymis. The epididymis continues into the ductus deferens. This is a microscopic image of the testes, the histological image of the testes, in which you can see the capsule, connective tissue capsule, thickening of capsule in the posterior part called as mediastinum testes, septas dividing the parenchyma, the lobules containing the seminiferous tubule, the seminiferous tubule having two types, epithelium having two types of cells, SERTOLI cells and the spermatogenic cells. This is a cut section of a seminiferous tubules having two types of cells, spermatogenic cells and the SERTOLI cells. There is a small quiz for you. The retitestes is lined by, you have to answer, it is simple cuboidal epithelium, simple columnar epithelium, simple columnar ciliated epithelium, or the pseudo-stratified columnar ciliated epithelium. Another question, which of the followings are produced by the SERTOLI cells? Inhibin, androgen binding protein, testosterone, or the semen. The blood-testis barrier is formed by which of the following cells? Capillary endothelium, capillary basal lamina, Leydig cells, or the SERTOLI cells. Thank you, dear students. If you have any question, you can contact me on the Google Classroom. Allah Hafiz.